Okay, welcome back everyone. So we're now going into part 2 of session 2, and we're going to start off with a talk by Lumbani. So Lumbani previously used to work at Reading in the Cocoa Group. He now works at the University of Cambridge. So Lumbani is going to talk to us about our Cocoa Soils project, which is a collaborative project with partners in Ghana. So I'll hand over to Lumbani.
Thank you. My presentation is on the GCRF Cocoa Soils project and is titled Remediation of Cocoa Soils in Ghana as a Route to More Sustainable Cocoa Production. This was a project that was implemented by a number of institutions. That's the University of Reading, Kwame Nkrumah University of Science and Technology, that's in Ghana, and then the Cocoa Research Institute of Ghana. As an introduction, I would like to mention that a decline in soil quality and its ability to sustain production through the crop's lifetime is a major challenge facing smallholder cocoa production. As you can see on the picture there, it's a productive farm, but with the years of production, soil fertility and quality can decline, which can lead to a decline in farm productivity. So in this 2-year GCRF project, we conducted a series of studies to investigate potential interventions to help improve soil quality in a sustainable way. And these studies were focused on the use of organic soil amendments, but we also had a study in which we looked at the effects of shade trees. So the laboratory studies were conducted at the University of Reading using soils imported from Ghana. And the greenhouse experiments on cocoa seedlings and the field experiments in mature farms were conducted by Craig, and also a socio-economic study in form of a survey was implemented by the Kwame Nkrumah University of Science and Technology. In this presentation, I'll focus on the laboratory experiments in which we looked at the effects of organic amendments on soil properties. Before I go into further details, I want to mention that the cocoa pod husks are a major farm waste in cocoa farms in Ghana and are rich in potassium and phosphorus, and therefore could be used to improve soil fertility. However, it is feared that when applied directly to soil, the cocoa pod husks, here abbreviated as CPH, can cause microbial nitrogen immobilization due to their high carbon to nitrogen ratio, while nutrients such as potassium can be lost through leaching. So here we assessed the effects of CPH residues and the CPH-derived compost and biochar soil amendments on the transformation and losses of carbon, nitrogen, and potassium in soil. Our hypothesis was that the transformation and losses of carbon, nitrogen, and potassium in soil would vary with application of CPH residues, compost, or biochar, and that compost and biochar application will improve soil properties such as nutrient availability. In these laboratory experiments, we used 2 soil types, that is a fellow soil and a neat soil, which were collected from different cocoa growing regions in Ghana. And these soils varied in their properties, for example, soil pH and the soil organic matter content. And we also used the 3 types of organic amendments derived from cocoa pod husks. That is the CPH residues, that is the raw cocoa pod husk crushed into small particles, and then biochar made from the cocoa pod husk, and also compost made from cocoa pod husk combined with poultry manure. And these 3 organic amendments also varied in their properties. For example, you can see here that the CPH residues had the highest C to N ratio, and the biochar here had the highest potassium content. And these organic amendments were applied at rates of equivalent to 10 tons per hectare for CPH residues and compost, and the 5 tons per hectare for biochar. And the experiments were conducted with or without 15N labeled urea fertilizer application. The fertilizer was applied at a rate equivalent to 20 kilograms per hectare for nitrogen. Here, just to show you the experimental systems, we conducted 2 microcosm experiments each of which lasted for a period of 4 weeks. In the first experiment, we measured different soil properties and the gaseous emissions from soil, whereas in the second experiment, the soil was enriched with water, and we measured different properties of the soil and the enriched samples. I will now show you some of the results from the laboratory studies, and I'll start with the soil incubation study in which we measured gaseous emissions. So for soil CO2 efflux, as you can see in this figure, it varied between soil types and organic amendments, and it was highest in the CPH residues treatment and lowest in the unamended soil, which is something that shows greater decomposition in the CPH residues treatment compared to the biochar and compost treatments, which is likely because of a greater quantity of biocarbon in the CPH residues treatment. What is not shown here is that urea application increased soil CO2 emission, the CPH residues treatment only. For nitrous oxide emission, it also varied between soil types and organic amendments, and it was highest in the compost treatment, whereas the biochar and the CPH residues treatment had the lowest nitrous oxide emission from soil. What is also not shown in this figure is that the nitrous oxide emission from soil increased with the urea fertilizer application. In the soil incubation study, we also measured microbial biomass carbon and the nitrogen at the end of the 4 weeks study period. I should mention here that the microbial biomass carbon is a good indicator of microbial biomass size. So what these two figures here show is that both microbial biomass carbon and microbial biomass nitrogen were highest in the CPH residues treatment, and they were lowest in the biochar and unamended soil treatments, which is something that indicates greater nitrogen immobilization with microbial growth in the CPH residues treatment. Now I'll move to the leaching study, starting with the leaching losses. For nitrate leaching, what this figure shows is that nitrate leaching was highest in the compost treatment and the lowest in the CPH residues treatment. The biochar treatment had the lower nitrate leaching compared with the unamended soil. What I should also mention here is that the urea fertilizer application did not significantly affect nitrate leaching. For ammonium leaching, it was also highest in the compost treatment and the lowest in the CPH residues treatment. But here we see that the ammonium leaching increased with the urea fertilizer application. After leaching the soils over a period of 4 weeks, we also measured how much urea fertilizer nitrogen was retained in the leached soils. So what this figure shows here is that there was a significant interaction effect of organic amendment and the soil type for urea N retention in the leached soils, which was driven by what happened in the neat soil, where there was a greater increase in the urea N retention in the CPH residues treatment. So this data also supports that there was a greater nitrogen immobilization in the CPH residues treatment. We also conducted the linear regression analysis, and this figure here shows that we found a positive relationship between the microbial biomass nitrogen and the urea nitrogen retention in the leached soils. So this is something that strongly suggests that the microbial immobilization of urea nitrogen increased with the microbial biomass size. This figure is for potassium leaching over the 4 weeks study period. So what you can see here is that all organic amendments increased potassium leaching compared with the an amended soil, but their effect varied between soil types. This is my final data slide, and I would like to show you the concentrations of soil ammonium, total nitrogen, and the potassium in soil at the end of the 4 weeks study period during which the soils were enriched with water. I should mention here that we found that the soil nitrate concentration was below the limit of detection in all treatments. However, for soil ammonium concentration, we found that the CPH residues treatment reduced soil ammonium concentration compared with all other treatments, which is indicating that the CPH residues had a negative impact on the soil available nitrogen. However, we found that all organic amendments increased soil total nitrogen and the potassium compared with the unamended soil. And the largest increase in the soil total nitrogen was found in the compost treatment, whereas the largest increase in the soil potassium was found in the biochar treatment. Now I'll go through a summary in form of conclusions derived from the laboratory experiments. First, the cocoa pod husks residues increased soil CO2 emission, which was possibly due to the rapid release of labile carbon from the cocoa pod husks residues, which increased microbial activity. And the cocoa pod husk compost increased soil nitrous oxide emission, which was likely due to its high nitrogen content and its release in soil increasing substrates for nitrous oxide production. Both the carbon dioxide and the nitrous oxide emissions varied between soil types and organic amendments, and the cocoa pod husk biochar treatment was associated with the low CO2 and N2O emissions. The cocoa pod husk residues showed the highest increase in soil microbial biomass carbon and microbial biomass nitrogen, the lowest nitrogen leaching, the lowest soil ammonium concentration after leaching, and the highest urea fertilizer nitrogen retention in the leached soils, which is something that indicates high microbial biomass size and nitrogen immobilization and reduced soil available nitrogen and reduced, as well as reduced nitrogen leaching in this treatment. All cocoa pod husk derived soil organic amendments increased soil potassium and total nitrogen compared with unamended control soil. And the highest increase in the soil potassium was found in the biochar treatment, whereas the highest increased soil total nitrogen after leaching was found in the compost treatment. Therefore, different forms of cocoa pod husk derived organic soil amendments can be used to improve specific soil properties. In one of my first slides in this presentation, I mentioned that field trials were established in Ghana and that they are continuing. Here I would like to mention that the additional funding through a GCRF Agritech project enabled implementation of a data capture system in these field trials, which was developed by Chris. And this allows more efficient data capture using mobile phones to scan barcodes on the trees and enter the observed data, and then the transfer of the observed data to a portal accessible by project partners from wherever they are. And this additional funding also enabled installation of weather stations covering the ecological zones in Ghana in which the field trials are located. To conclude my presentation, I would like to thank technical staff at the University of Reading, Craig, and the KNUST for the role they played in this work, and also acknowledge funding. And also, I would like to say thank you for listening.
Okay, thank you, Lumbani. And just a reminder that if people have questions, if you can put them into the chat, please. So moving on next, Jim Dunwell is going to talk about molecular aspects of cadmium accumulation in cacao. So I will hand over to Jim for the next presentation.
Hello, today I will describe some of the recent studies conducted at Reading on the subject of cadmium uptake in cacao. These were principally conducted by Isanola, the co-author here. Many of you will know the background to this problem, in that there is an acute problem of cadmium in some cacao or cocoa products, and this has led to potential problems in exporting, particularly into Europe. There have been many suggested approaches involving modified agronomy methods, but there are other options to address this, and these include genetic approaches and the role of cadmium transporters. Previous work conducted here had identified some of the candidate genes, including cacao NRAMP5, a transporter of cadmium, and a potential target for modification. As part of a more recent study, we've investigated a number of the target genes, including NRAMP, that encode proteins responsible for the uptake and translocation of cadmium. To do this, we selected 3 groups of clones known to accumulate cadmium at differing levels, and these are described in the table below. They come from work both published in Trinidad and from previous studies here conducted at Reading. From each of these clones, we selected and completed the NRAMP and HMA3 genes. They were amplified and sequenced and used to identify any genetic variation within these genes. I'll describe just a small proportion of the sequence data that we obtained, since time is limited today. Initially, we concentrated on genetic variation in the NRAMP5 gene. A range of sequence variations was identified. These were principally in the form of SNPs, that's single nucleotide variations, which were both present in some clones and not others. There were synonymous and non-synonymous SNPs. None of them were SNPs within the regions of the protein that is known to bind the substrates, but nevertheless we decided to investigate them in a functional test. These functional assays were used using a transgenic yeast assay system to test whether the various cacao genes were able to transport cadmium into the yeast cell. This is a quick and relatively simple means of testing the efficacy of the different variants of the target genes. The panels will show, and the background to this is that if there is no growth of yeast, it means that cadmium has accumulated in the cell, and therefore the functionality of the transporter gene is being proven. As shown here, there was no difference between the various forms of NRAMP5, which contain the various SNPs from the different subgroups of clones. A similar investigation of the HMA3 gene revealed a range of SNPs as with NRAMPs, but also transcript variants. In other words, some of the transcripts were truncated, shorter than the full-length gene, and therefore likely to encode a truncated protein. We also tested these in assays that I'll show later. The similar yeast growth assay of the cells containing the different alleles of the cacao HMA3 genes showed very high sensitivity in all cases, so that they were functionally active. However, the splice variants showed complete loss of function, except for one variant that encodes a truncated 750 amino acid peptide. In a more complex study shown here with a greater range of genes, other variants were tested. The overall results described here followed the results obtained previously. In summary, we can see that as the cadmium level in the culture medium increases from 0 up to 20 micromolar, the growth of the colonies on the plates decreases, but the rate of decrease differs between the genes shown here. So we see, for example, the confirmation of the previous NRAMP experiments, which grows at control and low levels, but then at 1 micromolar and above, there's no growth of the yeast, thus showing that expression of that transporter allows accumulation of cadmium in the yeast cells and therefore inhibits growth. Noticeably, but not linked to the present study, there is one gene from a completely different plant poplar here, a metallothionein gene, which shows complete resistance to cadmium right the way up to the highest concentrations. So there are other potential genes about which are not transporters, but ones which might actually bind at very high affinity the cadmium in the cell and stop it being toxic, because if it's bound within the cytoplasm or the ER, then it's not available to inhibit the metabolism of that cell. So there may be some other approaches that we could follow based upon those results. I'd now like to turn to another related series of experiments that we've been doing with collaborators, principally in Imperial College in London, and these have been conducted over the last several years and are still ongoing. These are designed to complement the molecular approaches that I described earlier, and they involve the exploitation of the fact that cadmium is known to exist in a range of different isotopes, and the use of those isotopes and the differential profile of them is something that's been used to examine cadmium uptake in other crops. Normally the cadmium is described as being of either heavy or light isotopes in two groups, and it's the ratio between heavy and light that is a distinct pattern, and it differs between plants, between parts of plants, and between the different crops that I've described. It's been shown that wheat and barley show similar patterns of isotope profile, and these differ particularly from plants which are known as hyperaccumulators that can accommodate and survive under much higher concentrations of heavy metals. The work I'll describe is the first example of the application of these isotope profiles, looking at the uptake into cacao and the genetic variation that exists for these patterns of uptake. In summary, in this particular element of the study, we used samples that were taken from 19 cacao clones grown in hydroponics under controlled conditions in the growth cabinets here at Reading. We found genetic variation to exist between these clones, both in the amount and overall accumulation of cadmium, and the partitioning between shoots and roots, and also in the isotope profile of those different component parts of the plant. We also then used the yeast system to examine the isotope profile in transgenic yeast cells, expressing the same range of genes from cacao as we tested in their overall uptake previously. We showed that there was genetic variation for each element of this whole process. We found that transgenic yeasts that express NRAMs had isotopically lighter cadmium than in cacao. We also found that the cadmium isotope signature of transgenic yeast expressing the HMA proteins suggests that they may contribute to cadmium sequestration. And overall, the data that we produced here is the first example of testing functionally transporter proteins in vivo using isotope fractionation studies. I'll show you one or two of the quantitative findings from this study in the next few slides. There's quite a lot of detail in this particular figure, but there are a few important take-home messages that I'll point out. The results here and the graph that you can see has cadmium concentration on the x-axis, and then the differential ratio of heavy to light isotopes on the y-axis here. The higher the value here, the heavier the isotopes that are present within the samples. And so this shows several important things. That within the roots, which are the black circles here, the concentrations tend to be much higher, up to 800 micrograms per gram, compared with the leaves here. This is not surprising, but what is interesting is the genetic variation that exists with each spot representing a particular clone. So there's a fourfold difference in concentration in this system between the low accumulators and the high accumulators. And also the leaves show systematic enrichment of the isotopically heavy cadmium isotopes relative to the whole plant. So there's differential genetic variation in each element of this process, both in the uptake and then in the partitioning between roots and leaves after uptake. If we look at a similar diagram representing data for the transgenic yeast, remember these are yeast cells into which we've introduced a range of the variant forms of the transporters that we've mentioned previously. The overall pattern is quite distinct, with the N-ramp, the 5 transgenics, so those are the squares here, showing a higher concentration of cadmium overall, but also a lower proportion of heavy isotopes. So it's higher in this axis, but lower in this axis. And this is true whether you introduce the N-ramp gene alone or in combination with HMAs or in other variants. So there's a particularly crucial functional introduction and activity of the N-ramp compared with some of the other transporters here. One of the questions often asked is how realistic is it to use a yeast system in this way? Obviously, ideally, you'd like to generate an equivalent number of transgenic cacao plants themselves, but to date, obviously, that's not possible. But one of the questions is to do with the localization of the introduced transporters in the yeast and whether their intracellular location is the same as that in the plant from which the gene was taken. So we used a conventional transgenic yeast approach, but linking a fluorescent marker, which is either green or red, to the introduced genes. So this is a green fluorescence linked to the N-ramp transporter here. This is a red fluorescence linked to a gene known to be expressed and produce a protein in the apoplast. So in other words, the N-ramp is located to the same location as a plasma membrane protein, and they overlap completely here. So the location of the introduced gene and its expression is at a cellular level is the same as we hoped and predicted. So it doesn't have a different location in yeast than it does in cacao. So in summary, we've provided the foundation of a more extensive survey of at least 2 of the target gene families, the N-ramps and HMA3 genes in cacao. We've shown that there's good evidence and distinct evidence of different genetic sequence variation between these genes, between the different clones, and also splice variation that exists, something that has not been previously described in cacao. We've also shown that we can use cadmium isotopes in quite a precise way to investigate the complex detail of cadmium uptake and translocation and its genetic components. However, like much of science, the more we do, the more complex we see the picture is. And so we have still got quite a long way to go before we understand the full details of the cadmium uptake process in this species and how this might inform genetic approaches in the future, whether we can investigate, well, we intend to investigate that with plans that we have at present. So thank you for your attention, and I'll stop there.
Okay, thank you, Jim. So if you have any questions for Jim, if you could please put them into the chat. So finally this afternoon, our final speaker is Jeremey Spencer. So Jeremey is a member of the Department of Food and Nutritional Sciences here at the University of Reading, and he's going to talk to us today about vascular and brain impact of cocoa flavanol intake. So I will hand over to you, Jeremey.
Thank you, Andrew. I'll just share my screen. Okay, hopefully everyone can see that. So first of all, thank you for inviting me today. I've found it very interesting so far. It's not my area at all, so I've been struggling with some of the details. Hopefully you won't find the same problem with what I'm going to present to you today, which is a little bit of a change of focus, but is looking at some of the human data for the benefits of consuming, particularly the flavanol component of cocoa, and in the context mainly of cognition, but also of the effects they have on the circulatory system in terms of our blood pressure and our blood flow. So first of all, just speaking more generally about flavonoids, I mean, as I'm sure all of you are aware, they're the plant secondary metabolites. We consume them as part of our diet. And over the last 20 years, I would say there's been a huge interest in their effect on various aspects of physiological function and disease prevention in humans. My main areas are their effects on the brain, particularly in the context of memory and learning and cognitive performance, but also traditionally from a vascular function point of view, in that when we feed these compounds to individuals that we recruit, we see reductions in blood pressure and we see improved endothelial function. In other words, improved blood flow around the body. And that's always regarded in a medical sense as being beneficial in reducing your risk of having a heart attack or a stroke, or perhaps falling into dementia in later life. So one of the first studies that was done in this area was more of an observational trial, and it was looking at the diet, the intake of these compounds. In this case, it was total flavonoid intake. I'll get onto the specifics of cocoa flavanols in a bit, but before I start, I just wanted to show you that over a 10-year period, those individuals that were consuming high amounts of flavonoids within their diet compared to low amounts had a much better cognitive evolution. And that meant that they were less likely to drop on something called the MMSE trial, which is the mini mental state examination, which is the type of test a neurologist will give to someone if you're worried about your memory and you're worried about perhaps getting Alzheimer's disease. So having these compounds in your diet daily on a regular basis can certainly protect the brain against decline in its functionality. And more recently, we've been interested in trying to get an idea about what might be going on a little bit more specifically in terms of which aspects of the brain are being affected most notably. The problem with the MMSE is it measures total brain function. And if you want to look at more specific outcomes like executive function, which is sort of a measure of attention and your ability to maintain accurate attention on tasks, then you need to design some more specific tests. And the next slide, or the next two slides, show you some examples of that. So about 10 years ago now, I had a BBSLC grant where we wanted to test the impact of cocoa flavanol supplementation acutely and chronically on cognitive performance. And we had 103 healthy 65 to 80-year-olds. At this time, they were all male, but we have done females more recently. And they're all cognitively healthy at baseline, so they weren't suffering from any diagnosed dementia. The data I'll show you in the next slide is the results of the acute consumption. So this after only 2 hours. And the cocoa extract we used was provided to us very kindly by Mars, and that was a benefit to us because they were also able to make a placebo for us. That's very important for cocoa in the context of cognition because, of course, you've got methylxanthines in there, and these definitely need to be matched, particularly in the context of an acute trial. So overall, our intervention, which was a cocoa extract, which was encapsulated, contained around about 500 milligrams of total cocoa flavanols. And this was divided into about 90 milligrams of epicatechin, 21 catechin, and the rest were the oligomers of epicatechin, catechin within there. Everything else was matched, as I say, including methylxanthines. So we carried out a huge array of cognitive tests, and this was in collaboration with psychology department at Reading. And we had 5 measures of executive function, which broadly were looking at the ability of someone to sustain accurate attention on complex tasks, and also episodic memory, which includes things like facial recognition, which you've been presented in minutes and then have to recall at a later date. And in both these cases, there were improvements in the group which consumed cocoa flavanols. I should say it's a crossover trial, so everyone took both of these. But you can see from a collapsed Z-score of all 5 tests, you could see there was a significant improvement in sustained attention after the consumption of the cocoa flavanols compared to the control. And also an attenuation of the decline, and this is a well-reported decline in episodic memory over a 2-hour period. Again, this decline wouldn't happen necessarily in normal day-to-day life, but only if you're actually cognitively engaged in a complex task. So there is some improvement seen here in the context of cognitive fatigue as well. So it might seem quite remarkable that such a group of compounds can have such an effect, but I mean, I'm a biochemist by background, so I've always had an interest in mechanism as well. And so over the last sort of 10 to 15 years, we've been looking at how flavonoids generally, and I would say actually the strongest data in this area is with flavanols, and more importantly, cocoa flavanols. There's important funding reasons for that, which I'm sure some of you are very aware of. But we're very indebted to these companies that are interested in the basic science of these compounds. So what I'm mainly going to show you today are the effect of these compounds on the peripheral and the cerebrovascular circulatory system in the context of those improvements in cognition which we observed. We've also seen their ability to inhibit neuroinflammation and also to modulate synaptic plasticity, which is, I mean, as 2 Nobel Prizes have shown, is one of the models of how memory is laid down within the brain. So there's a whole host of evidence, and I'm obviously going to concentrate on cocoa today, but there's also other flavanol-rich sources, like, for example, green tea, and even those that are found in blueberry, which have shown the effect of flavanol intake on lowering blood pressure in people with elevated blood pressure or hypertensive individuals, but also in healthy people where there is no high blood pressure. But you can also measure an improvement in vascular function, an improvement in blood flow to the peripheral tissues, but also to the brain. And I'll show you some data for that later. And of course, all of these are likely to not only have an effect on cognition, but also to reduce someone's cardiovascular disease risk. And actually on that point, I show you, this is data which came out in the last few weeks. This is the data from the Cosmos trial, which was run in the United States, which we were involved with. And this was the largest ever clinical trial to test the cardiovascular benefits of specifically cocoa flavanols. There have been lots of smaller trials up until this point, but nothing in the region of 21,000 individuals aged 60 and above, and followed over a 5-year period. And actually, as you will see on the left-hand side here, the endpoints were actually cardiovascular disease mortality. And so not only risk markers, but actually reduction of disease and death from cardiovascular disease. Now, I won't go into all of the complexities of this, but the 2 columns represent some clinical features which I don't have time to go into today. But for our purposes, the most important column is this second column, because this is only including the individuals that admitted to taking all of their cocoa flavanol supplementation over this 5-year period. And you can see there were significant reductions in all-cause cardiovascular mortality by 15%. And if you only looked at death from cardiovascular disease, there was an almost 40% reduction in death from cardiovascular disease over this 5 years compared to a placebo. So really exciting results. And if you look at the next slide, you can see where I've compared here the results of the Cosmos trial with the Mediterranean diet, and with saturated fat intake reduction and also sodium reduction or salt reduction within the body. And this shows really that cocoa flavanols are as powerful, if not more powerful, than the Mediterranean diet in its totality, or the reduction in saturated fat intake, or indeed the lowering of salt in reducing cardiovascular disease. So this, you know, there is really something, there is really an effect there. And there's definitely an interest in the mechanisms. So I guess what we wanted to know really was, were these blood flow changes which were underlying those effects on cardiovascular? So we can measure vascular responses to cocoa flavanol intake in the peripheral vascular system, so everywhere outside of the brain, and also within the brain. You can see over a 4-hour period, you have a peak at around about 2 hours. So this is an upwards movement on FMD, or an upwards movement on cerebral vascular blood flow indicate an improvement in your circulatory system, in your circulatory function. These will underlie or prevent increases in blood pressure over time. And we were very excited that these changes also occur within the brain. Incidentally, there were also reductions in blood pressure in this cohort. So I guess what my main question was whether or not these types of effects are mediated through the nitric oxide system, because we know nitric oxide production is intimately involved with changes in blood flow and reductions in blood pressure, and whether or not these changes could underpin actually what's going on when we have these compounds within our diets. I won't spend too long on this, but just the principle really is we know that if people consume flavanols, we can get enhanced cognition. If we use a blocker of our proposed mechanisms of action, then we should see a reduction in those effects. And this was a BBSRC grant that completed around about 2019. And the data show quite nicely that the first two bars here, this is the control, and this is the flavonoid, the flavanol intake. And you see the same increase in functionality and the same reduction in or attenuation in decline of functionality or memory performance. If you've then co-administered the nitric oxide inhibitor, then you partially reduce these effects. So there is a partial involvement of the nitric oxide system and nitric oxide bioavailability in the functionality of these compounds when they've been absorbed and when they've been consumed orally. And we also looked at this in the context of our blood flow precisely. So this was obviously on cognition itself. And this was on blood flow in the periphery first, measured by FMD. So again, we see nothing with our placebo. We see a nice increase, which slowly declines over a 6-hour period with the flavonoid treatment. And if you've used the inhibitor, you partially attenuate, block this rise in, or this improvement, enhancement in blood flow, again indicating that there is an involvement of the nitric oxide system in the effect that they have on the brain. You also see a reduction very strongly at 1 hour, less so at 2 hours. It wasn't quite significant here, but nevertheless, increase in cerebrovascular blood flow over a 2-hour period, and a blocking of this effect if you use the inhibitor, which blocks the system independently of the intake. So the last thing I just want to say is, so cerebral blood flow is a sort of composite measure of blood in and blood out, and that's interesting, but it doesn't tell us much about what type of effects they might be having on the brain. So we used a technique called resting state arterial spin labeling in the MRI at Reading. We had a cacao flavanol intervention versus a control. And this image here is a composite image of the baseline to 2 hours with the control removed, statistically removed. So this is only the effect of the cacao flavanols on blood flow within the brain. And you can see actually that these changes in blood flow are quite precise. And one of these regions is the anterior cingulate cortex, which is certainly involved in many of the types of cognitive tests that I mentioned earlier and that we were measuring improvements with. And actually, if you look at it a little bit more specifically, here's the ACC within the part of the frontal lobe of the brain. And the ACC has long been known to be involved in all sorts of executive control and attention and decision making and impulse control. And so it's not really surprising that if you increase blood flow to these regions, you're going to see some changes in these cognitive outcomes. But actually, I suppose more interesting than that is that the ACC is also quite important for the autonomic control of heart rate and blood pressure. And we know independently from what we've done in other intervention trials that these compounds, these cacao flavanols, also have an effect on heart rate and on blood pressure and on reducing blood pressure specifically. This has definite implications for aging because as people age, blood flow to this region is reduced. And actually, this is not the only region of the brain where you would suffer a loss of blood flow to a region independent of disease with the aging process. Another is the hippocampus, for instance, which affects short-term memory. So if we can restore some blood flow to regions of the brain which are reduced with aging, then we can restore functionality to older people, and that's exactly the type of effect that we saw when we were doing our intervention trials at Reading. So in summary, cacao flavanol intervention has now been shown to enhance cognitive function, executive function, and someone's ability to sustain their attention on a complex task. And it does this. I've shown today that it does this acutely, and we've also shown that you can get some chronic effects of that. So if you consume them over many weeks or many months. We've shown that this does involve partially the eNOS system and perhaps even nNOS within the brain. So there's definitely some functionality of nitric oxide. And thereby, you know, it's not the whole story, but it's certainly a part of the story. And then lastly, the enhancement of cognition and potentially these vascular changes that we see appear to be being mediated through the anterior cingulate cortex, which is quite interesting. I think there's been similar studies shown, very similar to the UoR, very similar types of responses within the hippocampus, and those are of course very interesting for Alzheimer's disease populations as well. So lots of excitement around cacao flavanols at the moment, particularly in light of the Cosmos trial. And if you are interested in anything to do with the types of benefits of consumption of cacao flavanols, then I would really recommend you have a look at the Cosmos trial website, which has a lot of the data summarized there. So lastly, I'd just like to thank colleagues in psychology and of course PDRAs and PhDs that have worked on this over the years. Most of the research was sponsored by the BBSRC or the MRC, and where we've done interventions with cacao flavanols, we've been very lucky to be able to work with Mars, usually as an industrial partner on these grants, who have been able to supply us with the interventions, which are, as I indicated at the beginning, essential when you're trying to do something with a complex food, if you like, or a complex extract of a food, because you need very nice control. So I'll stop there, and I'm happy to answer any questions that you might have. Thank you.
Okay, thank you very much, Jeremy, for a really fascinating talk there. So we're now going to go on to questions and answers. So a few questions have come in. So I've got a question here for Jeremy. Do we know if there is any genetic variation in the levels or types of flavanols in cocoa or in other crops?
I don't know the answer to that, but I think it would be very interesting to look at. I would imagine there would be. I mean, with flavonoids in crops, usually these things, so I understand it from crops like wheat and oats, and perhaps even tea, it seems to be, if anything, more affected by where it's grown and what's the intensity of UV exposure. And I know many of you might be aware of those types of experiments that were done as part of the ReLU project quite a long time ago now, where they exposed to full UV or partially blocked UV, showing that there were very different levels of these compounds which were being generated by the plant. So I don't know specifically, but it would be very interesting to look at that. And there have even been questions around where also the location of those in other plant tissues as well, because you know, as well as the cocoa pod and the seeds within, whether or not they're also laid down in other areas of the plant.
Okay, thank you. So another question to Jeremy again, from Uma over in Trinidad. So what is the best form to consume cocoa flavanols? Is it chocolates, cocoa-based beverages, or purified flavanols?
Yeah, well, as you can imagine, when I give these types of talks, most people ask the chocolate question. The problem with promoting these compounds within chocolate is because chocolate is usually quite high in fat and high in sugar, and they have their own negative effects on the vascular system. So you probably want to avoid obtaining them through chocolate if you can. Cocoa is perhaps better. Also, another aspect to take into account is the level of those compounds as well. I mean, I didn't show you here, but we've done separate experiments where around about 0.5 milligrams of epicatechin, 0.5 milligrams of epicatechin per kilogram body weight is the amount you want. So if you can get that into chocolate, if you can get that into cocoa, then you're certainly going to be expecting to have a vascular effect. I would say probably cocoa is better than chocolate just because of that sort of fat sugar problem. But yeah, at the moment, it's a big question about whether we should be promoting that because it's similar to the sort of red wine story. You know, people overindulge in things that they think are going to be beneficial. So we need to be careful with our messages, I think.
Okay, thank you. So, Jeremy, it looks like you're very popular today. So another question to you. What is it about cocoa flavanols that makes these particularly effective? Do the same compounds in other foods have the same effect?
They, yeah, so you do get some of those effects with blueberries, and people believe that, you know, this is mainly due to anthocyanins. But what they often forget is that, you know, those pathways for flavanol anthocyanin production are quite similar, or part of the same pathway. And actually, a lot of these fruits contain flavanols in addition to some of these other compounds. We don't know the reason why flavanols are more effective than others, but I would say out of all of the compounds that have been tested, it certainly seems like minus epicatechin is the strongest and most efficacious in what I've shown. Also, cocoa, unlike some other fruits which contain these compounds, maybe that's not really true when we look at sort of apple skin, but cocoa is known to contain the longer oligomers, and they're not absorbed in the upper GI tract, and so they start to be broken down and modified by the gut microbiota in a very similar way to fiber, actually. And so we've shown that not only do they get broken down by those gut microbiota, but the microbiota change in a beneficial way in the process of doing that. So it could be that, for example, cocoa flavanols, because they contain those longer oligomers, do have a better overall effect on our biology than, say, a smaller group of anthocyanins, which might be found in berries. I suppose what you're looking for is that sort of astringency or tannic structure within the mouth, which you do get with cocoa, and that's going to provide you with those vascular effects, but also some effects on the gut microbiota, which are now known to have their own effect on the blood-brain or the gut-brain axis, for instance.
Okay, thank you very much.
Okay.
So it looks like that's all the questions we've got. I would say if anybody thinks of any other questions to any of our speakers today, then please do feel free to drop us a line at any point. So finally, I'm going to hand over back to Paul Hadley now for a final wrap up.
Thanks, Andrew. So just a very few words from me. First of all, I'd like to thank the cocoa team, and particularly Emily, Andreas, Philippe, and Jeremy for helping us make this webinar such a success. And of course, I'd like to thank all the attendees for staying on for what was quite a long afternoon. And of course, for some of you, the timing of these webinars can be a bit challenging. So thank you all for attending. And as Andrew says, if any of you got any further questions to any of us, and it doesn't necessarily confine itself to the subject of the webinar, do get in touch with us. We'll be very happy to respond. For those who perhaps weren't able to attend that you know might be interested in the webinar, we are going to try to get a video of the webinar online as soon as we possibly can. It just remains for me to thank you all for attending, and hopefully we'll see you all soon at the International Cocoa Research Conference in December. Thank you very much. Bye then.